Okay, thanks very much for the introduction. It's an absolute pleasure to be here, and thank you for the invitation to come and share some thoughts and some of our experiences with you. As you can probably tell from my accent, I'm not a German national, but I have been working in Heidelberg for the past 5 years or so. My main research focus is on childhood brain tumours, but in the last 18 months to 2 years, I've been increasingly involved with trying to work out how we can bring the latest cutting-edge technologies into clinical application much more rapidly than we were doing previously. So the question that I was asked to address was, will genomic research improve treatment and outcomes in childhood cancer? And I think you can probably guess that the short answer from me is yes. Otherwise, I probably wouldn't be standing here talking to you. And I think it's important to realize and remember that actually childhood cancer as a whole is something of a success story if you look back historically. So treatment and survival have already improved dramatically over the past few decades. And in many cases, the treatments that have been developed through research and through clinical trials and through more focus on these entities have dramatically improved outcomes. But unfortunately, we know that there are some entities, particularly after relapse or progression of the tumour, where prognosis remains very poor. So this is a selection of the entities that we're now focusing on within the particular project that I'm going to speak about, where once that tumour comes back, once you relapse, unfortunately the outcomes for these children are still very bad. But these are the patients and the types of cancer where we have the biggest potential to make a difference by taking a new approach. And we think that a new type of strategy is now needed in the management and the treatment of these children. So at the moment, the traditional chemotherapies that represent standard of care are really targeting all rapidly dividing cells, which is why you have side effects such as hair falling out. And it's kind of a one-size-fits-all approach. So we don't really look in detail at the particular genetic alterations or what it is that's causing an individual's cancer. We just take the sort of oversimplistic view that all cancers of the same type are probably more or less likely to respond similarly to some of these drugs. But increasingly now, the approach that we're trying to take is what's using more targeted therapies, which have a lot of potential to act more specifically on cancer cells and try to spare some of that damage to the surrounding normal tissue. And it really doesn't mean that these new drugs are dramatically more effective at killing all cancer cells or that there are no side effects of these targeted approaches. But there are certainly some very clear examples now of where if you get the drug matching to a particular genetic alteration in a tumour, they can have a very, very good effect and they work very well. So the challenge that we're facing, just as not all people are the same, I think incidentally this one over here is maybe supposed to be me, so I'll leave you, make your own impression of that. We also know that not all cancers are the same. So as I said, my focus is on brain tumour research, and just within central nervous system cancer, there are around about 100 different types of tumour that go to make up brain cancer. So if you translate that to the rest of the body, it's an extremely complex problem that we're facing, and we really can't continue to use this one-size-fits-all approach. It's just not going to work for the children where these treatments are not currently working. So the solution that people are starting to use now and that we're trying to bring in Germany is this idea of personalized medicine. So using the very latest cutting-edge molecular profiling techniques to really understand more about the very precise details of what is causing one individual's cancer rather than looking at the whole population as a homogeneous mass. And by doing that, we hope that we can then find groups of patients, even sometimes very small groups of patients, who might have particular alterations in those tumours that make them sensitive to some of the new drugs that are being developed within the pharmaceutical industry and within academia. And this has really been made possible by a technological revolution that's really sort of dramatically increased over the past 5 to 6 years. So when the first human genome was sequenced, that project began in 1990, and 13 years later, the first draft sequence was announced, and that project in total cost around about $3 billion. And you can see now that it's really an exponential growth in how we can produce this data. So from when that project started back in 1990, we've moved on tremendously in terms of the technology being applied. And at the same time, the costs of those technologies have been decreasing even faster. And now we're at the point where instead of taking 13 years, a whole genome can be sequenced in 2 to 3 days, and it costs less than $5,000 to be able to do that. So it's really been absolutely transformational, the changes that have been going on in the technology that we're using now. So I just want to spend the second part of this going into a little bit more detail about the project that we're running in Germany. It's called the INFORM project, which is Individualized Therapy for Relapse Malignancies in Childhood. And this is a project which is running across the whole of Germany, so any of the pediatric oncology centers can submit samples to this project. And it's really for these children with progressive and relapse tumors where there's no other current standard of care option which is likely to make a dramatic improvement. So we're really focusing on this subpopulation of patients where we hope that we might be able to make some kind of real difference. And this is just to give you a brief introduction to what we're doing. So the samples come into a central pathology lab from across the country. We then take quite a comprehensive approach to how we're characterizing these tumors. So it's really using the very latest technologies, and I'll go into these in a little bit more detail on the next slide. And after that, there's a molecular tumor board, so people with very diverse experience from the clinicians, biologists, pharmacologists, and people who have very complementary expertise in all of the different segments of this pipeline come together to assess this data on a very individual level to try and see whether there's anything in that data about that individual's tumor which points towards a novel therapeutic strategy. So this RNA-seq component is looking for changes in the dosage of genes. So maybe you get too much expression of a gene which is driving the cancer, or you have too little of the gene creating protein that normally acts to block cancer. And we also look for what's called fusion genes, which is two completely independent genes which normally have nothing to do with each other. Sometimes they get rearranged and half of each one gets stuck together. And those can take on a new function which goes towards driving that cancer. We also do what's called exome sequencing, which is looking for errors in the DNA code that go to make up our genes. And we now know that sometimes just one single change in one letter out of the 3,000 million bases in the DNA can be enough to really dramatically change the behavior of a cell and turn it into a cancer cell. We also do low coverage whole genome sequencing, so we look across the whole genome to see whether instead of having the normal 2 copies of every gene, sometimes you get additional copies within the cancer cell, which again goes to driving that tumor, or you might lose a gene which would normally act to prevent the cancer from forming. And then we have 2 other complementary technologies in there to confirm the original diagnosis, and again, just to look for some other types of mechanism. And we think that only by taking a really comprehensive approach can you capture the wide diversity of different alterations which can cause cancer. So, as I say, all cancers, even within one particular type, are very far from being uniform. There are a huge number of different alterations and changes which can cause a normal cell to start proliferating too much and become a cancer. So you have to really take a very comprehensive approach to how we're profiling these and able to in order to be able to capture that diversity in what can go wrong. And this gives you an idea of the timeline for this process. So we start from a new biopsy of the recurrent tumour. So we require that we actually have tumour material from the current disease within each patient, because we know that when the tumour comes back, it can actually be quite different to the cancer that was there before it's relapsed. So we really need to make sure that we're profiling the current disease and trying to treat the current disease rather than what was there months or years previously. And then it goes into the lab where we extract the DNA and RNA, we perform these molecular analyses, and then the board meets to discuss that data and see whether there's anything in there that we think makes sense as a drug target. And our target is, from this rebiopsy to having the first report back, is less than 28 days, and at the moment we're hitting about 22 days for getting that turned around. And for the scale of the analysis that we're doing here, we think that's actually a pretty impressive timescale, and it's certainly within the range of time that it really can allow us to modify the treatment decisions and the treatment planning for the individual patients once that tumour relapses. So this is the progress that we've made at the moment. Since October 2013 until this month, we've approached 96 patients to enroll on this scheme. 10 of them, for various reasons, were not eligible to go on to the further stages. So we had 86 patients who were fully enrolled. In 5 cases, by requiring that new material at relapse, either there was not sufficient tumour content in there to be able to perform the analysis, or the diagnosis was actually found to be different to what we were expecting, and on that basis they weren't eligible. So 81 patients have been sequenced, and it's a range of different entities. So a lot of brain tumours, high-grade glioma, medulloblastoma, but also Ewing sarcoma, some rhabdomyosarcomas, osteosarcoma, neuroblastoma. So it's quite a broad representation of the different types of cancer that you see in childhood. The leukemias are slightly underrepresented. That's because when we really restrict it to the highest risk subset of leukemias, that's fortunately a slightly smaller proportion of the population than first-line leukemia. So within these 81 patients where we've got the molecular data back, we found something within that data which we would consider there to be some evidence for considering a new treatment in around about half of the cases. And what we also do with that data is give it a score as to the strength of the evidence that it really makes sense to give that a possible new treatment. So sometimes we know, as I say, that one very precise alteration in the DNA code can really give a very, very strong rationale that a particular drug is very likely to work in that patient, and that would give a very high score for prioritization. And sometimes the evidence is not as strong, so we just see upregulation of a cancer pathway where there's a drug somewhere in that pathway, and it's not really as precisely targeted. So we're trying to understand about how the strength of that confidence can then translate into the responses that we see if a decision is really made to use those drugs. I just want to leave you with one of the success stories that we've had out of running this project. And of course, from those 70 or 80 patients, this is just one example that I'm picking out. And as I say, only in half of them did we find a target where we had some evidence to point towards a new therapy. And even amongst those where we did find a target, sometimes the treating physician decided that actually the treatment they were on was already showing some response and they didn't want to modify that at that point, or the access to the drug it was not possible to get hold of that drug for that patient for whatever reason. So the number of sort of really striking responses that we've had, it's certainly not every patient on whom we're doing this analysis, but we have seen some anecdotal examples where it really makes a very clear difference. And this is one example of a patient who was recruited quite early on within the process, who had what was at that stage an inoperable myofibroblastic sarcoma. This was a really very large tumor. And by performing the molecular analyses within this program, we identified one of these fusion genes that I was mentioned. So part of this gene known as ALK, which is a very well-known cancer gene, was stuck onto half of another gene from a completely different chromosome. And this rearrangement was causing this part of the ALK gene to be much more active than it normally is. And that was thought to be very likely that this was the key event in that tumor, which was really driving those cells to be malignant. And on the basis of this finding, we were able to recruit that patient onto a phase 1 clinical trial of a new ALK inhibitor that's being developed. And you can see here the response that had. So this is the baseline MRI scan. You can really see how large and this tumor was. It was on the neck and spreading up through the head. And after 6 weeks of being on this drug, we saw a really, really dramatic shrinkage of that tumor to the point where the surgeons were then happy to go in and say, actually now, I think I can operate on this without having too many adverse effects on that patient. So the surgeons were able to remove the rest of the tumor, and around about, as I say, this is one of our earliest patients. So 18 months later, fortunately, this teenage patient is now still in complete remission of their tumor. So this was, as I say, this is far from being the story for every one of the patients we've seen, but I think for us it was quite a nice example to show that the rationale behind what we're doing, at least in some cases, does make sense. So where we're hopefully going now is to move on from this being a registry study where we do the analyses and report that back to the treating physician, and then it's up to them whether they want to make use of that data to try and get hold of the new drugs. What we really want to do is turn it into a full phase 1-2 clinical trial where we don't really care what entity we're looking at, but we want to base it purely on the biology of the tumor. So rather than having a trial for a particular tumor type, we have several arms within a trial that are driven by the biology. So if you have a mutation in gene X, regardless of what type of tumor, you'll be recruited onto that arm of the clinical trial. And that's what we're now planning, with the hope that it will start within the next 12 to 18 months. And we've also started discussions with other centres across Europe now. It's been, for us, quite a steep learning curve, and there have been some barriers to really trying to get this type of project up and running. But now that we've got this experience, and there are some other centres who are taking similar approaches, what we want to try and do is to coordinate those approaches so that we're taking more of a Europe-wide integrated approach. People are working together instead of having lots of parallel efforts doing slightly similar but also a little bit different approaches. So just to sum up, we know absolutely for certain that every cancer is different, and just continuing to take a one-size-fits-all approach is not going to always be the best way in which to plan treatment, particularly when you've tried several other things already and unfortunately those haven't shown a marked response. The technology is now available that we can rapidly and relatively cost-effectively identify the alterations which are present in an individual's tumour on a per-patient basis. We know that targeted drugs, when they really precisely match their target, can be extremely effective, but equally we know that they're not going to be a magic bullet for all of the patients. For some of the genes, we know that that's what's causing the cancer, but there's just no drug available for that gene so far. Cancers also, it's becoming increasingly clear that if you just use single targeted drugs, they can quite quickly develop bypass mechanisms around that drug, so they become resistant to a single targeted agent. And that's why people are moving more towards combinations of drugs now to try to preempt those resistance mechanisms. And as we've heard already, access to some of these drugs, particularly for pediatric patients, remains quite a major challenge. And we do have some funding in place in parallel to this project now that allows the clinicians to be able to apply for money to purchase those drugs off-label, where it's an approved drug but not for the particular tumor type. We're trying to enable really to make it so that people can get access to those drugs, but still that can be a challenge if it's only in the trial phase in adults and there's no data in pediatric patients. But I think at least we do have reason for some cautious optimism that for some patients we can make a difference already by taking these approaches, and as we learn more, as the access to drugs gets better, we can really start to make more and more of a difference by bringing these approaches into the clinic. And as I say, this is really, sorry, this is the only slide which still has some German on it. Sorry about that. It's really a nationwide study. There's a huge amount of people involved in this team, and it is a collaborative effort. And I think that's part of the reason why we feel that it is working quite well at the moment, and that we've been able to get people to feel like they're involved in the project and have a stake in it, and it is something that they believe in as well as a team, not just as a series of individuals. So thank you very much.
Thanks, David. As this morning, if I could ask you to join a panel and we'll have some brief